scientists have used a pocket-sized DNA scanner to sequence the most complete human genome ever assembled with a single device. The 700 pounds, $1,000, portable gadget, which is no bigger than a mobile phone, could one day help us detect diseases in real time. The breakthrough brings us closer to the day when family doctors will order up genome scans during a regular checkup. Use Star Trek-style tricorders. Scroll down for video. We are definitely approaching the point where sequencing genomes will become a routine part of advanced clinical exams, lead author Professor Matthew Luce, a geneticist at the University of Nottingham, told AFP. The new sequencing method is the first to read long, unbroken strands of DNA, yielding a final result that is 99.88% accurate. The process of assembling a genome is like piecing together a jigsaw puzzle, said co-author Professor Nicholas Loman, an infections specialist at the University of Birmingham. The ability to produce extremely long sequencing reads is like finding very large pieces of the puzzle. Critically, the so-called nanopore technology sheds light on poorly understood regions of the genome governing the body's immune responses and tumor growth. This may help detect cancer DNA in the blood, and pick up tumors before they are symptomatic or visible through radiological techniques, said Professor Loman. In the case of a patient with a suspected infection, the sequencing can be used to uncover the invasive genome of a virus or bacteria, he told AFP. We could also simultaneously look at how the patient is responding to that infection, he added, noting that each individual's immune system is different. The same is true for sequencing a person's microbiome, the vast community of microbes we each host, mostly in the digestive tract. For personalized medicine, we will want to build up a picture of how individuals may respond to antibiotics and anti-cancer drugs, Professor Loman said. The human genome is composed of more than 3 billion pairs of building block molecules, and grouped into some 25,000 genes. It contains the codes and instructions that tell the body how to grow and develop, but flaws in the instructions can lead to disease. The first decoding of the human genome, completed in 2003, took 15 years, cost $3 billion, and marshaled hundreds of scientists and the computing power from 20 major universities and research institutes. The new sequencing, carried out by a dozen researchers and half a dozen MinION devices cost a few $1,000s and took 3 weeks to complete. In 5 to 10 years, genetic sequencing will be as ubiquitous as boiling a kettle or making a cup of tea, predicted co-author Professor Andrew Beggs, from the University of Birmingham, one of 9 institutions involved in the project. The researchers pieced together the genome by passing strands of DNA through minuscule tube-like structures, manufactured by Oxford Nanopore Technologies, along with electrically charged atoms. Changes in the electrical current identify DNA molecules, which can then be mapped. The compact biomolecule sequencer has minimal moving parts and plugs directly into a laptop or tablet, which supplies power and collects the sequencing data. Unlike terrestrial instruments whose sequencing runtimes can take days, this device's data is available in near real time. Analysis can begin within 10 to 15 minutes from the application of the sample. Currently, the device can only analyze purified DNA samples that have been prepared in a lab, meaning it cannot read a blood or saliva sample directly.